When we talk about the central nervous system, the spotlight almost always lands on the massive cerebral cortex.
Of course. That's where we think all the action is.
Right, the seat of memory, language, consciousness. We assume that's where the real complexity lies. And if you even think about the cerebellum, you probably boil it down to, what, one word?
Balance.
Balance.
And that, right there, is probably the greatest historical mistake in neuroscience.
I think so, too.
That structure, the one responsible for that tiny little job of balance, the cerebellum, is anything but simple. I mean, it is the brain's ultimate hidden control center.
It really is.
It contains an absolute majority of all the computing power in your head. It's responsible for far more intricate functional processing, exquisite timing, sequencing, and, we now know, even high-order cognition than we ever gave it credit for.
I find that historical neglect absolutely fascinating, especially when you look at the sheer numbers.
The numbers are staggering.
So today, we are undertaking an extremely thorough deep dive. This is a massive synthesis of some very dense neuroanatomical and physiological source material. Think of this as your shortcut to mastering a complex neuroanatomical chapter.
We're going to detail the architecture, the connections, and the revolutionary functional understanding of the cerebellum.
And our mission here is precision. We're going to try and map this structure completely.
We have to be precise because the architecture is just. It's relentless in its uniformity and its detail. We are not skipping a single thing here. What this structure is, how it's built, or how its immense network of connections gives it its unparalleled computational power.
Okay, so where do we start? We have to place it spatially first.
Yes. Let's start with location. The cerebellum is a key structure of the hindbrain. It originates from the primitive rhombencephalon, and it sits neatly tucked away in the posterior cranial fossa.
So if you imagine the brainstem, the pons and the medulla, the cerebellum is nestled right on the back of them, dorsal to them.
Exactly. It sits just beneath the massive occipital lobes of the cerebrum.
And it's separated from them by that fold of dura mater, the
Tentorium cerebelli.
Right. So it's effectively forming the roof of the fourth ventricle. It's an incredibly protected posterior location.
And this brings us immediately to the great paradox of the cerebellum.
The size versus the cell count.
Exactly. While it only accounts for about 1/10 the weight of the cerebrum, maybe 130 to 150 grams in adults, its cellular density is, well, it's nothing short of astonishing.
Let's really try to unpack that scale for a minute. If we estimate the entire central nervous system contains roughly, say, 100 billion neurons.
Which is a good working number.
The massive cerebral cortex, the part we think does all the work, accounts for about 20% of those. 20 billion neurons.
Which means the cerebellar cortex holds the remaining 80%.
80%.
We are talking about 80 billion neurons compressed into this small, relatively compact structure.
80 billion. That is 4 times the neuron count of the cerebrum, the part responsible for our language and consciousness, packed into 1/10 of the space. That fact alone should just shatter the historical view of the cerebellum as just balance. That sheer density demands some extreme folding.
It has to. It does. The massive cellular density explains why the surface of the cerebellum isn't just folded, it's highly convoluted. It's characterized by these numerous, tightly packed, curved transverse grooves.
Then those grooves separate the
folia, right?
The folia, which are these slender gyrus-like folds and the layers of varying depths. This is why when you look at a sagittal section, it looks like a beautiful branching tree.
The arbor vitae.
The arbor vitae, the tree of life. It reflects that immense need to pack surface area into minimal volume. And the cellular complexity, this is the functional foundation for its role as this superlative general-purpose processing engine. It's capable of handling everything from predicting when a baseball pitch will land to, sequencing a sentence.
Okay, so let's start with that big picture then. The macroanatomy. We need the framework that houses all those billions and billions of cells. When we view the cerebellum from the outside, what are the primary, most visible divisions? At the gross level, the framework is really tripartite. We have 2 very large, laterally placed cerebellar hemispheres.
The big wings on the side.
Exactly. And then running right down the midline, uniting these 2 hemispheres, is the vermis.
The vermis.
It's a raised structure, and its name derives from the Latin word for worm-like.
And it really does look like a midline worm connecting the 2 massive lateral wings.
It does. And because of this extremely high level of folding, the entire structure is riddled with these grooves and fissures that define the anatomical boundaries.
So we really need to detail the two major transverse fissures that structure the entire cerebellum. Let's start with the first one.
The first one, and it's functionally very important, is the primary fissure.
Okay.
This fissure is a deep groove that runs transversely across the superior surface of the cerebellum. And it's crucial because it separates the cerebellum into two main sections.
Front and the back.
The front and the back. The anterior lobe situated in front, and the very large posterior lobe situated behind it. And that split often reflects the older parts, the anterior versus the newer parts, the posterior, functionally speaking.
So the primary fissure gives us that main front-to-back separation of the main body of the cerebellum. What about the other, perhaps developmentally older, major division?
That would be the posterolateral fissure.
Okay.
It's also called the flocculonodular fissure.
Right.
And this one is significant from a phylogenetic standpoint because it is the earliest fissure to appear during fetal development. It sits inferiorly and posteriorly, separating the small, evolutionarily ancient flocculonodular lobe from the rest of the main cerebellar mass.
And what do we call that main mass?
The rest of it, the anterior and posterior lobes together, are collectively termed the corpus cerebelli.
That developmental history is such a powerful clue. I mean, the structures that appear first often control the most fundamental processes, right? Like breathing, balance. And the flocculonodular lobe, the first to separate, is indeed the seat of vestibular function.
It is, exactly.
Yeah.
And just by observing the exterior shape, we can describe the surfaces. The superior surface, it slopes gently laterally and posteriorly, and the vermis is really clearly visible here.
But the inferior surface is totally different.
Dramatically different. It's characterized by a massive enlargement of the cerebellar hemispheres, which have expanded greatly in primates and, of course, in humans.
And how are those large inferior hemispheres separated?
They're separated from each other medially by a really deep cleft or valley, which we call the vallecula cerebelli.
The vallecula.
And the median area, that inferior surface, which is the vermis, is mostly concealed. It's tucked down deep into the depth of that vallecula, unlike how visible it is on the superior surface. Hashtag 1.2, the trilobar architecture, the three main lobes.
Okay, so based on those two major fissures, the primary and the posterolateral, we arrive at the three foundational anatomical divisions of the cerebellum. Let's just delineate those three traditional lobes clearly.
Right. Based on those fissures, the three traditional lobes are: one, the anterior lobe. It lies in front of the primary fissure. And as we'll see, this is largely responsible for basic trunk and limb sensory information.
Got it.
Two, the massive posterior lobe situated between the primary fissure and the posterolateral fissure. This is the lobe that has undergone the greatest expansion in humans.
And this is where all the cognitive stuff happens.
This is where the territories responsible for high-order thought are located. And then, three, the flocculonodular lobe.
The oldest one.
The oldest one. It lies posterior to the posterolateral fissure. And this lobe is comprised of two parts: the nodule, which is the vermis component, and the flocculus, which is the hemisphere component.
So we have the anterior lobe, the massive posterior lobe, the great expansion of the cerebellum, and then the evolutionarily ancient flocculonodular lobe dedicated to balance. These three blocks really provide the main functional architecture.
They do.
But the complexity, I mean, it really jumps up when we zoom in on the specific folds, doesn't it?
It really does. To move from that high-level function to precise anatomical targets, which is absolutely essential for imaging and clinical diagnosis, we have to use the systematic nomenclature for the lobules.
This is where things get dense.
This is where the large cell system comes in. It provides a numerical designation, Roman numerals I through X, running sequentially from the most superior fold right down to the most inferior.
Okay, so this is where we need to slow down and really use this list of lobules to build a mental map for you, the listener, rather than just rapidly reciting a bunch of names. This highly specific anatomical address system applies to the midline vermis, I through X.
We begin at the very top, superiorly, with the lingula, which is lobule 1.
Lingula.
The lingula is functionally tied to the vestibular system, although it's tiny. Immediately following that is the central lobule, which covers 2 and 3.
Okay.
This is where we begin to integrate somatosensory information from the body. These are separated from the prominent, most superior hump of the cerebellum, the culmen.
The culmen, that's IV and V.
Phi and V. And the culmen is crucial because it contains a detailed map of the body, particularly the limbs.
So just to recap, lingula, central, culmen, that's I to V, and they occupy the space anterior to the primary fissure.
Correct.
So that makes up the entire anterior lobe. Now, what happens when we cross the primary fissure into that huge posterior lobe?
Once we cross that primary fissure, we enter the posterior lobe with the declive, which is lobule sixth.
The clive.
Following that are the enormous territories of the folium, which is VIA, and the tuber, which is VIIB.
So VIA and VIA, folium and tuber.
And these large VIA and VIIB territories represent the vast majority of the posterior vermis. Moving further down the lower parts, we encounter the pyramid, which is eighth, the uvula, which is IX, and finally the most inferior part, the nodule, which is X.
That is incredibly precise for the midline. Now, how do the surrounding hemisphere lobules align with that IX vermal address system?
So the hemisphere lobules are paired structures, but their naming becomes a bit more general, sometimes more complex, as you move superiorly, though they do align structurally. For instance, the hemisphere regions corresponding to the culmen 4 and V are often just vaguely grouped.
But the naming gets more refined as you go down.
Yes. Aligned with the declive, lobules a sixth, is the simple lobule, or HVI.
And then we get to the massive expanded human regions. This is where it gets interesting.
Yeah, this is where human evolution really shows its anatomical preference. The hemisphere territories that correspond to the folium VIA and the tuber VIIB are greatly enlarged.
What are they called?
They're referred to as the superior semilunar lobule, or HVAI, and the inferior semilunar lobule, HVIB.
Okay.
Below that, we find the baventer lobule, HV8, and the tonsil, HIX.
The tonsil is the clinically significant one, isn't it? Because it can herniate.
It is. In cases of increased intracranial pressure, it can herniate down through the foramen magnum, which is a critical medical emergency. And then finally, we have the flocculus, or HX, which corresponds to the nodule.
So that systematic IX nomenclature is the core map. But I know our source material really emphasizes the adaptation of this map that was necessary for humans and primates.
It does.
And this is the crucial link to modern function.
That modification is absolutely essential because it accommodates that massive enlargement of the lateral hemispheres, the neocerebellum. The Schmahmann and Lancer subdivisions, which were introduced around 1999 and 2000, designated specific regions like Kreis 7 and Kreis 6 within those large hemisphere lobules.
Specifically within HVIIA and HVIIB.
That's right.
Cruci and Cruz second. They almost sound like pilgrimage routes. Why was this new nomenclature so necessary?
Because these regions have expanded so vastly, they receive such massive input from the prefrontal and parietal cortices, the areas responsible for our most advanced cognitive skills, that the original IX vermal system just wasn't sufficient to capture the sheer volume and functional specificity of the human cerebellar cortex.
So Cruci and Cruz second are now considered the anatomical seats of it.
Cerebellar-mediated cognition, language, and executive function.
So the IX system gives us the universal map, and then terms like Crucer and 2 give us the major functional territories that modern neuroimaging uses. The practical clinical relevance for you, the listener, is that fMRI studies use these exact lobule systems to connect specific anatomical addresses to clinical functions, to disease states, to abstract tasks. If a neurosurgeon or researcher knows a function happens in Crucerin, they know exactly which neurons are likely responsible. It's an incredible address system.
So to execute any function, those 80 billion neurons have to communicate with the rest of the nervous system.
Of course.
And that is achieved via 3 pairs of massive fiber bundles called the cerebellar peduncles. These structures are basically huge cables that anchor the cerebellum to the brainstem.
And they frame that characteristic diamond shape of the floor of the fourth ventricle.
They do.
So we need to analyze each peduncle as a major information pipeline, defining its direction of travel and its primary cargo. Let's start with the superior cerebellar peduncle.
Okay. The superior cerebellar peduncle, or the brachium conjunctivum, connects the cerebellum rostrally, so it's traveling up toward the midbrain.
Oh.
Its primary functional mandate is to carry efferent fibers.
So output.
It's the primary output. These fibers originate from the deep cerebellar nuclei, specifically the dentate, emboliform, and globose nuclei. And these fibers are destined for targets like the red nucleus and, critically, the thalamus, which then relays them up to the motor cortex.
So if the DCN is the command center, the superior peduncle is the primary output route for all the motor and cognitive plans. Does it carry only output?
No, it's primarily output, but it does contain some afferent fibers, some input.
Okay, what kind?
These input fibers originate from the rostral spinal cord, specifically the ventral spinocerebellar tract, which carries unconscious proprioceptive information. It also carries afferents from the trigeminal nucleus, which informs the cerebellum about sensory input from the face.
But its main job is to take process commands out.
Always remember its main job is output.
Got it. Superior peduncle primarily takes information out. Now for the middle one, that's the biggest one, I believe.
That's the middle cerebellar peduncle, the brachium pontis, and it is architecturally immense. It connects the cerebellum transversely to the pons.
And it's almost all input.
Overwhelmingly. This peduncle is composed of afferent fibers input, carrying an enormous volume of information derived predominantly from the cerebral cortex. This is the anatomical link between the cerebrum and the cerebrocerebellum.
And this information, it originates in the cerebral cortex, descends, and then it synapses where before entering the cerebellum.
Synapses in the pontine nuclei. The cerebral cortex motor, premotor, sensory prefrontal areas sends massive projections down to the pontine nuclei.
And then
The pontine nuclei neurons then send their axons across the midline, forming this giant, dense bundle, the middle cerebellar peduncle, which terminates in the contralateral cerebellar hemispheres. This massive data dump is the foundation of the neocerebellum's ability to refine cognitive and motor plans.
wow. That massive input from the cerebral cortex via the middle peduncle is what defines the lateral hemispheres. Finally, the inferior cerebellar peduncle, which we know is complicated because it's structurally split.
The inferior cerebellar peduncle is the oldest and most complex. It's subdivided into the more lateral restiform body and the more medial juxtarestiform body.
Okay, let's break down the restiform body first.
The restiform body is primarily afferent, carrying crucial sensory and error signaling information.
What kind of information?
This includes proprioceptive data derived from the spinal cord, specifically the dorsal spinocerebellar tract and the cuniocerebellar tract.
So that's constantly telling the cerebellum the current status of the body's limbs and joints.
Constantly. And importantly, it also carries the climbing fibers originating from the inferior olivary nuclei,
The error signal.
the error signal.
So restiform body is contextual input and error signaling. What about the juxtarestiform body?
The juxtarestiform body is the mixed section and is primarily involved in equilibrium. It carries primary afferent mossy fibers that originate from the vestibular nerves and vestibular nuclei.
Crucial information for balance and head position.
Absolutely. Additionally, it contains the key efferent fibers from the vestigial nucleus of the DCN, projecting directly out to the vestibular nuclei.
So it's both input and output.
It's a mix. This combination of incoming vestibular data and outgoing vestigial command confirms its absolute priority is maintaining posture and equilibrium. It also contains projections from the reticular formation, further linking it to core brainstem regulatory centers.
All right, we've established the external framework and the communication highways. Now we have to venture inward, past that convoluted cortex, to the white matter core. This is where all those incoming and outgoing wires meet. Hashtag tag 2.1, the white matter core and internal organization.
The white matter core is the dense structural foundation of the entire cerebellum. It lies immediately beneath the surface cortex. When you section it sagittally, it creates that recognizable, beautiful branching pattern that early anatomists termed the arbor vitae.
The tree of life. That term arbor vitae gives us such a great visualization of this structure. It looks like a massive inverted tree with branches extending into every single fold.
Precisely. The white matter core extends into the numerous folds, the folia, as these thin laminae, like the pages of a book radiating from a central spine.
And it's composed of what exactly?
It's composed of a dense traffic jam of axons. These include the two massive influxes of afferent fibers, the mossy and climbing fibers bringing information in, the axons of the Purkinje cells,
Which are the sole output of the cortex.
the sole efferent output of the cortex, and the excitatory axons of the deep cerebellar nuclei, which generate the final output. The white matter is the crucial junction box for all computation.
Okay, so deeply embedded within this white matter core are the crucial output centers, the deep cerebellar nuclei, or DCN. These nuclei are the indispensable link between the internal processing of the cortex and the rest of the brain. Without them, the cerebellum is functionally silent.
Completely silent. They are strategically located near the roof of the fourth ventricle. And the DCN are composed of two main populations of neurons. Large neurons with extensive dendritic trees, which are the main source of the output signal,
And what else?
and smaller neurons that receive direct excitatory input from the mossy fibers before the cortical loop even processes the information.
There are 4 pairs of these nuclei arranged in a really critical order from lateral to medial. And this arrangement, it mirrors the functional zones of the cortex right above them.
It does. Moving from the largest and most lateral inward, the 4 pairs are, first, The dentate nucleus,
A big one.
by far the largest and most prominent nucleus in the human brain. It has this distinctive, highly crumpled, purse-like appearance.
It's like a bag with an opening.
Exactly. And that opening is called the hilus, and it faces anteromedially. The dentate contains both large and small neurons, and it is the functional generator for the neocerebellum. It receives massive inhibitory input from the lateral hemispheres.
From Crest-Bruck and Crest-2.
That's right. So the dentate, receiving information from the largest, newest, most cognitively oriented part of the cortex, is naturally the largest output nucleus.
That makes perfect sense.
It is. Next, moving slightly medial to the dentate, we find the ovuliform nucleus. This is typically somewhat oval or wedge-shaped.
And next step.
Number 3, the globose nuclei. These are generally several small, irregularly shaped nuclear masses lying medial to the umbelliform nucleus.
And as we mentioned earlier, these two, umbelliform and globose, are often not treated separately in clinical conversation.
No, and that's vital context. Clinically, and sometimes functionally, they are grouped together and referred to as the interposed nuclei.
The interposed nuclei.
And this combined structure receives inhibitory input primarily from the perivermis region of the cortex.
So that's the intermediate zone of the spinocerebellum, responsible for skilled limb movement.
Correct. And then fourth, and most medial, is the vestigial nucleus.
Okay.
This is the most medial nucleus, located very close to the midline and near the roof of the fourth ventricle. And due to its medial position, it receives inhibitory input from the most medial cortical zones, the vermis and the flocculonodular lobe, the ancient vestibulocerebellum, hashtag 2.3 connections of the DCN.
Okay, the DCN is where the rubber meets the road. We have to solidify the core paradox of cerebellar processing.
Yes.
It's this precise inhibitory veto that's applied to a constantly excited command center. So what is the crucial overarching rule governing the connection between the cortex and the deep nuclei?
The crucial rule is this two-pronged output system. All processing within the cerebellar cortex culminates in one place, the Purkinje cell.
Right.
Purkinje cell axons are the sole effort pathway out of the cortex, and they project inhibitorily using the neurotransmitter GABA onto the DCN neurons.
They're always trying to silence the DCN. So if the entire complex 80 billion neuron cortex has one job, and that job is to inhibit, then what is the nature of the final output that's actually leaving the cerebellum?
The DCN neurons themselves are the final common pathway. They are the source of all cerebellar output to the rest of the brain, projecting via the peduncles.
And are they inhibitory too?
No. Unlike the cortex, the DCN neurons are inherently excitatory, utilizing glutamate. They are tonically active, they're always firing, because they receive constant, direct excitatory input from the incoming Mossian climbing fibers.
Okay, this is the core logic. So imagine the DCN neuron is a motor that's just running constantly. It gets excited by the incoming data.
Right.
The Purkinje cell is the regulator, or maybe a dimmer switch, constantly trying to turn that motor down or off based on the cortex's computation.
That's a perfect analogy. The final excitatory output signal leaving the DCN is therefore the result of that subtractive process. The ongoing tonic excitatory drive from the input fibers minus the precise, timely inhibitory control delivered by the Purkinje cell.
So the cerebellum functions by using its highly organized cortex to modulate an underlying, constantly excited command center.
Yes, and this requires a highly specific topographical organization to prevent signal mixing.
Does the cortical zone inhibit only its corresponding DCN?
Precisely. The organization is strictly parasagittal and topographically locked. The DCN receive inhibitory input from the cerebellar cortical areas directly above them in dedicated longitudinal bands.
Let's lay those out.
The dentate nucleus receives inhibition from the massive lateral hemispheres, the cerebrocerebellum. The interposed nuclei receive inhibition from the intermediate zone, the perivermis or spinocerebellum, and the vestigial nucleus receives inhibition from the medial zone, the vermis, and the flocculonodular lobe, the vestibulocerebellum.
So this means the cerebellum is organized into three almost totally independent vertical processing strips.
Yes.
And each is focused on a different functional goal balance, limb control, or planning, with basically no crosstalk at the output stage.
None at all. It's perfectly modular.
All right, let's zoom in now on the cortex itself. We've established this is where 80 billion neurons are crammed into this repetitive, almost crystalline structure. And this uniformity is what makes the cerebellum so functionally unique. Hashtag 3.1, the uniform architecture.
The uniformity is the defining architectural principle. The cerebellar cortex has a remarkably regular structure across every single folium and lobule.
From the ancient flocculonodular lobe to the newest parts of Crest II?
Everywhere. And this suggests that regardless of the data it's processing, a vestibular signal, a visual prediction, an abstract thought, it performs a fundamental, identical computational transformation everywhere.
So it's a universal processing chip built on a modular array.
Exactly. And this repetitive architecture is divided into the three visually distinct layers we see in all sections.
So from the outer surface inward toward the white matter core, what are they?
We find three layers. First, the outermost and most cell-sparse molecular layer, or ML. Second, the narrow central row of giants, the Purkinje cell layer, or PCL. And third, the innermost densely packed granule cell layer, or GRO. Hashtag tag 3.2, the Purkinje cell layer, PCL.
The PCL is the anatomical signature of the cerebellum. It contains those massive, unique output neurons.
The PCL consists of a single, highly organized row of Purkinje cell stomata. These cells are truly massive, measuring 50 to 70 micrometers in diameter.
That makes them some of the largest neurons in the brain.
They are. It's estimated we possess about 15 million of these gatekeepers in the human cerebellum, and they run the entire length of the folia like beads on a string.
And their input structure, the dendritic tree, is just legendary in neuroanatomy. Can you describe the sheer scale and the geometry of the structure?
The dendritic tree of a Purkinje cell is enormous. It spans 200 to 300 micrometers high and 250 to 300 micrometers wide. But the crucial geometric detail, and this is essential for understanding the computation, is that this tree is highly flattened. It's planar.
So if the folium is like a page in a book, the Purkinje cell dendrite is like a massive two-dimensional fan running across the page.
Exactly. This fan is rigorously oriented transversely across the folium. It runs perpendicular to the long axis of the folium and, crucially, perpendicular to the input fibers coming through the molecular layer.
That geometry is key to catching incoming signals.
It is. Furthermore, the capacity for input is immense. Each Purkinje cell dendrite is covered by approximately 175,000 spines, and those spines are the primary targets for the contextual input signal we'll discuss shortly.
So 15 million giant neurons, each covered on 175,000 potential input points, and their only function is to apply J-Bergic inhibitory control to the deep cerebellar nuclei. This is the ultimate regulatory system.
And their axons, before they deliver that inhibitory veto, they actually provide internal feedback. The PC axon descends to the white matter core, but gives off these recurrent collateral branches before terminating in the DCN.
And what do those do?
These collaterals are significant because they loop back to inhibit nearby Purkinje cell stomata and provide localized inhibitory control within the PCL itself, ensuring the network is tightly focused. Hashtag tag 3.3, the molecular layer, ML.
Okay, moving superficially, let's look at the molecular layer, the thick, fibrous outermost layer. It's cell-sparse, but it's dominated by synaptic connections.
The molecular layer is approximately 300 to 400 micrometers thick. Its contents are structural and cellular. Structurally, it's dominated by the vast planar dendritic trees of the underlying Purkinje cells and Golgi cells.
And what else?
It also contains the long, thin, parallel fibers, the axons of the granule cells, running longitudinally. And the cell bodies here belong to two key types of inhibitory interneurons, the basket cells and the stellate cells.
The interneurons are the local tuners, right? They sharpen the signal. Let's focus on the basket cells.
Basket cells are positioned in the deeper part of the molecular layer, right next to the PCL. Their axons run transversely, parallel to the PC dendritic plane.
And their genius is in their output.
It is. Their axons wrap around the stomata and the proximal axons of multiple Purkinje cells, forming a dense inhibitory basket structure.
So this delivers potent, localized inhibition.
It does. And it's crucial for the process of lateral inhibition. It ensures that if one PC is excited, its immediate neighbors are silenced.
That lateral inhibition is essential for defining the boundary of the excited zone, making the output incredibly precise. And the stellate cells.
Stellate cells are located more superficially. Their axons also provide inhibitory input, but they target the dendritic shafts and spines of the Purkinje cells, modulating the input received right at the source.
So it's a fine-tuning of the signal before it even reaches the PC soma.
Exactly. Hashtag tag 3.4, the granule cell layer, GCL.
And now we arrive at the density champion, the granule cell layer. This is the innermost layer and the most densely packed structure in the entire central nervous system.
It is breathtakingly dense. The GCL is about 200 to 300 micrometers thick. Its packed organization is why we cite that 80 billion neuron count. It contains approximately 2.4 million neurons per cubic millimeter in the human cortex.
And the primary neuron type driving this density is the tiny granule cell.
Granule cells, GCs, are the most numerous neuron type in the brain, period.
Right.
Estimates suggest an astonishing 2.7 to 3.5 trillion GCs in the human cerebellum.
Trillion.
Trillion, accounting for the vast majority of those 80 billion cerebellar neurons.
Wow. What is the path of the granule cell axon, and how does that translate into the functional input system?
The GC axon is the source of the contextual signal. It ascends vertically from the GCL, passes through the PCL, and enters the molecular layer where it performs its signature move.
The T-split.
It abruptly bifurcates splits into a perfect T shape to form the parallel fibers, PFs.
And those parallel fibers, the PFs, run the length of the folium, acting like these long sequential data lines.
They run longitudinally along the folium, parallel to the long axis of the folium, and therefore perpendicular to the planar dendritic fans of the Purkinje cells.
And that cross-orientation is the computational engine.
It is. PFs can travel huge distances, from 1 millimeter up to 10 millimeters, acting as a critical delay line.
So as one parallel fiber sweeps across the dendritic fan of a single Purkinje cell, it activates it at 175,000 different points. And because the PF is so long, it can sweep across the dendritic fans of thousands of PCs sequentially.
Yes, it distributes the mossy fiber signal widely, both spatially and temporally.
So that is the core mechanism of data dissemination.
It is. Each PF makes excitatory glutamatergic synapses onto approximately 175,000 PC spines, and it also excites the local interneurons basket, stellate, and Golgi cells to help modulate the signal.
Okay, beyond the granule cells, we have the other key interneurons in the GCL.
We have the Golgi cells. They're located in the superficial GCL. Their large dendritic trees ascend into the ML to receive input from parallel fibers.
And their axons.
Their axons are inhibitory, GABAeric, and they feed back directly onto the granule cells within the glomerulus. They are a crucial local inhibitory controller.
So they regulate how widely and how long the initial mossy fiber input is allowed to excite the granule cells.
Exactly.
And what about the less common, more specialized cells?
The source material also references the Lugaro cells, located just below the PCL. These are long, horizontally oriented cells thought to be inhibitory interneurons, receiving input from parallel fibers.
Okay.
More functionally specialized are the unipolar brush cells, or UBCs. These are found primarily in the flocculonodular lobe and ventral vermis, the ancient areas.
And what makes them unique?
UBCs are unique because they are highly excitatory interneurons that receive direct, robust input from mossy fibers, and then greatly amplify and feed that excitation onto the granule cells.
So this specialization in the vestibulocerebellum suggests a need for extremely rapid, high-gain processing for balance signals.
That's the thinking, yes. A need for very fast, very powerful amplification.
All right, now we fully transition to the data that is actually entering this crystalline structure. We know there are two major types of afferent fibers, and they speak the language of excitation.
That's right. Both major input systems, the mossy fibers and the climbing fibers, use glutamate as their primary neurotransmitter and are thus excitatory.
But we need to underline a critical point here.
Yes. Before either fiber type enters the cerebellar cortex to engage the Purkinje cells, they both exert a strong, direct excitatory control over the deep cerebellar nuclei neurons.
So they excite the output nuclei first.
First, this simultaneous direct excitation and indirect cortical regulation is the absolute foundation of the cerebellar loop.
Okay, so mossy fibers are the contextual input. They provide the bulk of the non-air information regarding what the body is doing and what the cerebrum is planning. Where do they originate?
MFs are the vast majority of afferent input. They originate from multiple, diverse sources throughout the CNS.
Like what?
Proprioceptive signals via the spinocerebellar tracts. Massive planned command data from the pontine nuclei.
Carrying the cerebral cortex's intentions.
Exactly. Also input from the reticular formation, sensory input from the face via the trigeminal nucleus, and critical information from the vestibular nuclei regarding gravity and acceleration.
And they terminate exclusively in the granule cell layer, but they don't just dump their information there, do they? They form a highly structured processing hub.
They do. They terminate in specialized structures known as the cerebellar glomerulus. This glomerulus is a microscopic pre-processing hub.
What are the components of this critical hub?
The glomerulus is a small synaptic island. It contains up to 400 total synapses. The main components are, 1, the large central mossy fiber terminal, which is excitatory.
Okay.
2, the dendritic claws of multiple surrounding granule cells, which are the excitatory recipients. And 3, the axons and dendrites of the Golgi cells.
The local inhibitory feedback controllers.
That's right.
So the mossy fiber releases its signal, the granule cells pick it up, but the Golgi cell is right there to immediately moderate the length and intensity of that signal.
That's the dynamic. The MF input is highly divergent. One MF terminal can excite 40 to 50 granule cells. This wide distribution of the contextual signal is essential, but it is constantly refined by that Golgi cell inhibitory feedback loop.
So if the input is too long or too intense,
The Golgi cell inhibits the granule cell, limiting the context spread. Overall, MFs convey continuous, varied information related to the body's current state, environment, and movement context.
so if mossy fibers are the broad, constant stream of information, climbing fibers are the lightning strike of the error signal, and they are uniquely singular in their origin.
They have only one known source in the entire CNS, the inferior olivary nucleus, or ION, located deep in the medulla oblongata.
That solitary origin means that the error signal is perfectly wired and controlled,
Perfectly.
and their termination point is what gives them their name and their power.
CFs terminate exclusively and powerfully on the dendritic tree of the Purkinje cells. The relationship is typically simplified to 1-to-1. One climbing fiber innervates one Purkinje cell.
Although it can branch a little bit.
It may branch slightly to innervate up to, say, 10 PCs locally, but the principle holds.
Can you describe the physical relationship and the resulting electrical signal?
The CF literally wraps around the PC dendrite. It climbs it, hence the name, and makes an enormous number of contacts, approximately 1,000 to 1,500 synaptic contacts.
1,000 to 1,500.
Yes. And this massive input results in an extremely potent, reliable, and sustained high-frequency burst of activation in the Purkinje cell, which we know is the complex spike.
And that is functionally distinct from the simple spike we hear about.
Oh, absolutely. The simple spike is the graded, continuous, low-frequency firing that's generated by the parallel fiber contextual input. The complex spike, generated by the climbing fiber, is a massive, sudden, all-or-nothing burst.
And the complex spike is the specific signal for error.
It is. It means there was a mismatch between intention and outcome, and it temporarily silences the PC's simple spike firing, signaling an event that requires learning and adjustment.
Chemically, beyond just the potency, what defines climbing fibers?
They primarily use the excitatory amino acids glutamate and aspartate, but they're also rich in modulatory substances like acetylcholine, dopamine, and calcitonin gene-related peptide, or CGRP.
So that suggests that the error signal isn't just an electrical spike.
No, it's a profound chemical event that causes long-lasting functional changes in the Purkinje cell, enabling plasticity.
And finally, their structural organization defines the entire architecture of the cerebellum.
The CF inputs, the olivocerebellar projections, show a strict, highly organized mediolateral parasagittal organization. They're wired in narrow, adjacent strips or bands.
And this creates distinct functional zones.
It does, often mapped using markers like zebrin, creating these alternating zebrin-positive and zebrin-negative bands.
Labeled C1, C2, D1, D2, and so on.
And these zones map precisely onto the output targets in the deep cerebellar nuclei, defining the computational structure of the entire cortex. The climbing fiber pathway is the anatomical blueprint for cerebellar function.
Okay, we have all the component pieces. Let's assemble them into the core computational loop, defining how the cerebellum uses these parts to refine and optimize every single output.
So the process starts with the necessary redundancy of the inputs,
Okay.
the input stage. Both mossy fibers and climbing fibers enter the cerebellum and immediately, simultaneously, excite the deep cerebellar nuclei neurons directly.
So this provides a constant, tonic, excitatory background drive. The DCN is always ready to fire.
Always ready.
The DCN is excited, and now the cortex has to process the incoming data and provide its regulatory veto.
Right. Cortical processing. First, the mossy fibers deliver contextual data to the granule cells. Then the granule cells project that context via the long parallel fibers.
The PFs sweep across the PC dendritic fans, delivering the graded, continuous.
The simple spike input. And then, if an error occurs, the climbing fibers deliver the potent, selective, complex spike input, which temporarily overrides the simple spike.
So the Purkinje cell is integrating this constant contextual flow against momentary error flashes. What is the PC's job in the output stage?
The PC's job is the veto. It acts as the regulator, continuously trying to suppress the DCN. This sustained, powerful inhibition is the entire purpose of the 80 billion neurons.
And the beauty of the circuit is that subtractive output.
Exactly. Final output. The final command leaving the cerebellum generated by the DCN neuron's firing rate is the tonic excitatory drive received directly from the MFs and CFs, minus the precise, temporally regulated inhibitory control delivered by the Purkinje cell.
So the output is a fine-tuned, time-adjusted signal that has minimized the perceived error.
That's the goal. Hashtag 5.2 intracortical modulation and timing, the interneurons.
The interneurons are the local editors, then, ensuring that the veto signal is not only accurate but highly focused.
The basket and stellate cells are responsible for spatial refinement through lateral inhibition.
How does that work again?
By inhibiting the PCs adjacent to the most active PC, they create a narrow, highly defined population of active PCs. Think of it like taking a flashlight beam and focusing it down to a laser point.
And this sharpening ensures the inhibitory signal targets a very specific set of DCN neurons, increasing the precision of the output.
It does.
And the Golgi cells, they regulate the quality of the input data before it even reaches the PC.
Golgi cells provide that crucial inhibitory feedback within the glomerulus. This loop helps modulate the duration and intensity of the initial mossy fiber granule cell activation.
So it's effectively controlling how far and how long the contextual information is allowed to spread via the parallel fibers.
Yes, and this is vital for maintaining temporal accuracy.
Which brings us to the parallel fiber system acting as a timing device.
This is one of the most widely accepted theories of cerebellar computation. Since the PS are extremely long and run transversely, the excitation delivered by a single mossy fiber input doesn't reach all Purkinje cells simultaneously.
Right, it takes time to travel.
It does. Instead, the signal propagates slowly along the PF, creating a specific temporal sequence of activation across many PCs.
And this mechanism is believed to be crucial for the cerebellum's ability to time complex motor sequences, anticipate movement, and sequence cognitive processes.
It's a precisely calibrated delay line.
Okay, so if the cerebellum is the ultimate editor, it must be able to change its script. This ability to learn and refine skills motor learning requires synaptic change or plasticity.
Yeah.
What are the key cellular mechanisms?
The primary site of learning is the synapse between the parallel fiber and the Purkinje cell spine. And the central mechanism is long-term depression, or LTD.
We hear about LTD far more than LTP in the cerebellum. Why is that?
LTD is considered the fundamental mechanism for motor learning because it is explicitly error-driven.
Okay.
LTD is a weakening of the synapse. It occurs specifically when the Purkinje cell receives near-simultaneous input from two sources, the parallel fiber providing the predictive context, and the climbing fiber delivering the unexpected error signal.
So, the climbing fiber is essentially telling the Purkinje cell, hey, the context you just predicted via that parallel fiber was wrong. Reduce your sensitivity to that pattern next time.
Precisely. This simultaneous activity registers a mismatch, leading the PC to reduce its responsiveness to that specific pattern of parallel fiber input in the future.
And by weakening an inhibitory input,
By weakening its own input, the PC allows the DCN to fire differently during that context in the future, thereby refining the output and minimizing the error.
but learning also requires strengthening. Is long-term potentiation, or LTP, also observed?
Yes. While LTD weakens connections, LTP strengthens them. LTP is observed at the parallel fiber Purkinje cell synapse when specific stimulation protocols are used, particularly when the parallel fiber is active without the concurrent error signal from the climbing fiber.
So a successful prediction gets reinforced.
Exactly. It suggests a reciprocal relationship to LTD is necessary. The circuit must be able to both correct by weakening incorrect patterns with LTD and reinforce successful non-error patterns with LTP. This balance ensures adaptive and lasting learning.
All right, now let's connect that cellular logic back to the macro framework. The three traditional anatomical lobes align almost perfectly with the three major functional divisions of the cerebellum, based on evolution and connectivity. Hashtag 6.1, the historical and phylogenetic divisions.
These historical divisions provide a really clean functional breakdown, from ancient survival to modern cognition. First is the archicerebellum, or the vestibulocerebellum,
The oldest.
the oldest evolutionarily conserved part. It encompasses the flocculonodular lobe and adjacent inferior vermal parts, lingula I, uvula I's, nodule X.
And its input.
Primarily afferents from the vestibular apparatus, the inner ear, and the secondary vestibular nuclei, delivered via the inferior peduncle, specifically the juxtarostiform body.
And its output is through.
It projects via the most medial output center, the fastigial nucleus, to the vestibular and reticular nuclei.
So its function is all about
Maintenance of equilibrium, axial and proximal limb movements, and critical gaze stabilization, like the vestibulo-ocular reflex. It's the constant stabilizer.
Okay. Number two?
2 is the paleocerebellum, or spinocerebellum. This includes the rest of the vermis and the immediately adjacent paravermal or intermediate regions.
And this covers mostly the anterior lobe.
Primarily the anterior lobe, lobules I through V, and parts of the posterior lobe, 6 and 8. Its input is extensive, detailed cutaneous and kinesthetic afferents from the spinal cord via the spinocerebellar tracts. It has a detailed map of the body.
Output.
It projects via the interposed nuclei, the globose and emboliform, and the fastigial nucleus to key descending motor systems, notably the red nucleus and the reticular formation.
And its function is primarily movement execution.
Yes, specifically controlling limb and trunk musculature, maintaining postural tone, and performing online correction of ongoing movement. It's comparing what was intended versus what is actually happening moment to moment.
And finally, the big one.
3, the neocerebellum, also called the pontocerebellum or cerebrocerebellum. This is the massive, evolutionarily newest structure comprising the vast lateral hemispheres, Crucis I and Crucis II.
Input is from the cerebral cortex.
Overwhelmingly, massive input from the entire cerebral cortex via the pontine nuclei. Its output projects via the largest output center, the dentate nucleus, to the ventrolateral thalamus, which then relays widely to the motor, premotor, prefrontal, and parietal cortices.
And its function is motor planning, motor learning,
And, crucially, higher cognitive and affective functions. Hashtag 6.2, the longitudinal functional zones, the zonal arrangement.
So while the phylogenetic division is useful, the true functional map, as defined by those climbing fibers, is not transverse but longitudinal. It's organized into functional strips.
This is the critical anatomical realization. The cerebellum functions not in horizontal lobes, but in 3 distinct longitudinal parasagittal stripes, which correlate perfectly with their output nuclei.
Let's run through them.
First, the medial zone, the vermis. This stripe projects exclusively to the fastigial nucleus.
And it controls axial and proximal control, balance, eye movements.
Yes. It influences the core stability centers of the brainstem. Second, the intermediate zone, the paravermis. This stripe projects to the interposed nuclei.
And this controls the distal limb musculature and the execution of highly skilled, learned movements.
Correct. Influencing the rubrospinal and reticular tracts. And third, the lateral zone, the hemispheres. This massive stripe projects to the dentate nucleus.
So it influences abstract processing, motor planning, and cognition via those extensive loops back to the cerebral cortex.
That's the one.
This longitudinal strip organization really emphasizes that the cerebellum is highly modular. Each strip is performing the same basic computation context versus error, but applying it to a completely different type of input.
Whether it's balance, movement, or thought. Hashtag 6.3, detailed afferent systems. Source-specific inputs.
So we have to look at the source-specific inputs in more detail because the function of the zone depends entirely on the information it receives.
Absolutely. Starting with the most rapid and ancient, the vestibulocerebellar mossy fibers.
Where are they from?
They originate from the primary vestibular nerves, from the inner ear, and the secondary vestibular nuclei in the brainstem. They terminate specifically in the flocculonodular lobe and the ventral vermis.
So they are the instantaneous signal for gravity and movement, essential for stabilizing gaze and posture.
Yes. Next, the detailed sensory map input from the body, the spinocerebellar and trigeminocerebellar mossy fibers.
And these carry.
These fibers carry detailed somatotopic information, a map of the body surface and muscle status, about muscle stretch, joint position, and skin input.
And they target the vermis and paravermis.
Yes, of the anterior lobe and parts of the posterior lobe. For example, the dorsal spinocerebellar tract carries proprioceptive information from the trunk and lower limb, while the cuniocerebellar tract carries the same information specifically from the upper limb.
This precise mapping allows the spinocerebellum to constantly monitor and correct limb trajectories.
Constantly.
And finally, the vast system that defines the neocerebellum, the corticopontocerebellar projections. This is the path abstract ideas take to get edited.
This pathway is monumental. The fibers originate from massive areas of the cerebral cortex, not just the motor and somatosensory areas, but overwhelmingly from the high-order association areas.
So prefrontal, parietal, cingulate cortices.
All of them. These fibers descend, pass through the internal capsule and cerebral peduncle, and then synapse in the pontine nuclei in the brainstem.
And from there,
the pontine nuclei then generate massive mossy fibers that cross the midline and enter the cerebellum via the middle cerebellar peduncle, terminating overwhelmingly in the lateral hemispheres, Crucis I and Crucis II.
That input density from the prefrontal and parietal cortex is the anatomical basis for the cognitive revolution in cerebellar research.
It is the definitive proof. Our source material emphasizes that cortical areas involved in planning, logic, sequencing, cognitive tasks. They project heavily to the dentate nucleus territories, specifically the D2 zone.
This massive pathway links the most complex aspects of human thought to the cerebellum's precise computational engine.
It does. Hashtag 6.4 climbing fiber afferents from the inferior olive.
We know the inferior olivary nucleus provides the error signal, but the ION itself has functionally segregated subdivisions, right? This ensures the error signal is delivered to the exact zone that needs correction.
It does. The ion N is divided into the principal, medial, and dorsal accessory olivary nuclei, and their projections are specific, following the longitudinal functional zones.
Let's lay out that map.
The dorsal accessory olive, or DAO, projects predominantly to the vermis and medial parts of the cortex.
The balance and axial control zone.
Right. The medial accessory olive, or MAO, projects mainly to the intermediate or paravermal parts.
The limb control zone.
And the principal olive, or PO, projects mainly to the massive lateral hemispheres, crista est and crista seca.
The planning and cognitive zone. This is truly phenomenal anatomical organization. The error signal for fine limb control comes from a different part of the ION than the error signal for abstract planing.
And each error signal is wired to its specific functional strip of Purkinje cells.
And this organization is what creates those precise parasagittal bands we talked about, the C1, C2, D1, D2 bands.
It is. Each band is defined by its climbing fiber input from a specific segment of the ION, ensuring that the cortical processing zone, the error signal, and the deep nucleus output are all locked together in a closed-loop system for high computational specificity.
All right, the computation is complete. The Purkinje cell has applied its inhibitory veto, and the DCN is generating the final excitatory command. This output now has to exit the cerebellum and influence the brain's motor and cognitive machinery. Hashtag 7.1 vestigial nucleus output, medial zone.
So the vestigial nucleus receiving input from the medial vermis responsible for posture is focused on core stability. Its output fibers primarily exit the cerebellum via the inferior cerebellar peduncle.
The juxtarestiform body, specifically.
Right. However, some fibers take a longer route called the uncinate tract, hooking around to exit through the superior peduncle.
And where do these core stability commands go?
They target the vestibular nuclei and the reticular formation in the pons and medulla. They also influence midbrain centers responsible for eye movements.
So functionally, this path regulates what?
It regulates axial motor control, core stability, equilibrium, and gaze stabilization. And critically, these projections are mostly ipsilateral.
They stay on the same side.
They do. Since the motor pathways influenced by the vestibular and reticular tracts typically descend without a complete midline crossing, the vestigial nucleus influences the same side of the body that its associated cortical zone received input from. Hashtag 7.2 interposed nuclei output, emboliform globose intermediate zone.
The output for the intermediate zone, the limb control system, is much more centralized.
Their output is exclusively routed via the superior cerebellar peduncle. And here is the key anatomical feature for limb control. This output pathway immediately decussates.
It crosses the midline in the midbrain.
Immediately.
So the output from the right intermediate zone crosses to the left side of the brainstem. Where does it terminate?
It terminates at two main locations. The magnocellular part of the red nucleus in the midbrain.
Which is the origin of the rubrospinal tract controlling distal limb movement.
Correct. And various parts of the thalamus,
Right.
specifically the ventral lateral and ventral posterior lateral nuclei.
And these thalamic relays then project to the motor cortex and related motor planning areas.
They do.
So this is the classic cross-control pathway.
It is. The cerebellum is designed to correct the motor output of the contralateral cerebral cortex. By having the superior peduncle output cross the midline, the intermediate zone on one side influences the distal limb movements on the same side of the body.
Because the ultimate descending motor pathways, like the corticospinal tract, cross back over lower down.
It's a beautifully engineered double-cross system. Hashtag 7.3 lateral nucleus output, dentate nucleus lateral zone.
And finally, the dentate nucleus, the output generator for the cerebellum and cognition.
Like the interposed nuclei, the dentate output is also exclusively via the superior cerebellar peduncle, and also immediately decussates across the midline in the midbrain.
But unlike the interposed nuclei, the dentate's targets are far more expansive, which reflects its role in high-level planning.
Its primary targets are the thalamic nuclei, the ventrolateral, the ventral anterior, and the centromedian nuclei.
And critically, the output from these thalamic centers then projects widely to nearly the entire cerebral cortex.
The entire cortex. This includes M1 and premotor areas, but the projections extend aggressively to the prefrontal, parietal, and association areas.
That broad projection confirms, beyond a shadow of a doubt, that the lateral cerebellum is not just optimizing movement, but optimizing thought.
The structural proof is undeniable. The dentate output is associated with motor planning, sequencing, and the highest cognitive functions. Our source material highlights a crucial anatomical segregation within the nucleus itself.
What's that?
The ventral dentate nucleus specifically projects to the M1 and premotor areas, the motor functions, while the dorsal dentate nucleus projects almost exclusively to the parietal and prefrontal association areas.
The cognitive functions.
This anatomical split perfectly reflects the division between motor optimization and cognitive optimization.
For centuries, the function of the cerebellum was just a clinical black box. We could only deduce it by the devastating, clumsy effects of damage. Modern imaging has given us the tools to watch those 80 billion neurons at work in real time. Hashtag 8.1, functional mapping via modern imaging.
Functional imaging techniques, particularly fMRI and PT studies, have been transformative. They've allowed us to correlate specific tasks with activity in precise anatomical addresses, really fulfilling the promise of the Larsell system.
And what do these studies confirm about the classic motor execution zones?
Motor execution tasks, like tapping a finger, walking, repetitive movements, they show activity focused primarily in the evolutionarily older areas, the anterior lobe, lobules IV, and the posterior vermis, lobule 8th.
Which aligns perfectly with the spinocerebellum's known function of correcting and executing ongoing movement.
Perfectly.
And what about the evidence for the higher order roles?
This is where the neocerebellum just shines. Cognitive tasks, including working memory, generating word lists, performing spatial tasks like navigation, even tasks involving emotional processing, show strong, consistent activation in the massively expanded lateral hemispheres.
Specifically CRIS4, CREST7, and VIIB.
Exactly. This confirms the cerebrocerebellum's deep functional integration into abstract, non-motor processes.
So imaging proves that the cerebellum is interconnected with the cerebrum via these widespread, parallel, closed feedback loops. It's a general processor.
It is. The functional map shows that the cerebellum is influencing not just how we move, but how we think. The system uses the same computational logic context versus error, regulated by the Purkinje cell, to refine any system it connects to.
So if the loop is motor, the output is smooth movement.
And if the loop is cognitive, from the dentate to the prefrontal cortex, the output is refined executive function, focused attention, or faster thought sequencing. Hashtag 8.2, the triad of clinical ataxia. The so what?
So understanding this structure is crucial for interpreting pathology. The primary manifestation of cerebellar dysfunction is ataxia.
A lack of voluntary coordination, characterized by errors in range, force, and timing.
And historically, the clinical view is dominated by the classic cerebellar motor syndrome.
Yes, exemplified by the work of Holmes back in 1939. This syndrome describes classic motor deficits resulting from lesions to the intermediate or medial zones.
Symptoms include what?
Generalized instability, dysmetria, a classic sign, the inability to judge the distance or range of a movement, leading to undershooting or overshooting, and an intention tremor.
So a tremor that is absent at rest but appears and worsens as the patient tries to reach for a target.
That's the one.
But the modern view, informed by the neocerebellum's expansion and the work of Schmahmann, acknowledges the full triad of dysfunction.
Correct. The modern framework recognizes a broader spectrum, often referred to as the Schmahmann syndrome, which emphasizes three concurrent areas of deficit.
What are they?
First, the cerebellar motor syndrome, the traditional ataxia of gait, limbs, and speech. And second, the cerebellar cognitive affective syndrome, or CCAS.
This is the non-motor manifestation.
It is typically seen with damage to the lateral hemispheres, quiz of the A2, and the dentate nucleus. CCAS includes profound disturbances in executive function planning, working memory, spatial cognition, language production deficits.
Like what kind of language deficits?
Like dysprosody, which is an abnormal rhythm and inflection of speech, and agrammatism, or difficulty using grammar correctly. And finally, affective regulation issues, exhibiting either blunted affect or, conversely, disinhibition.
The profound importance for our learner, for you, is that understanding the structural organization, the DCN output, the longitudinal zones, allows for incredibly accurate predictive diagnosis.
It is absolutely essential. Knowing the zonal arrangement means a clinician can predict symptoms based on lesion location. Damage confined to the medial vermis vestigial nucleus pathway. That primarily results in truncal ataxia and balance issues, often without significant limb involvement.
And damage to the intermediate zones.
That results in classic appendicular ataxia, limb clumsiness, dysmetria, and intention tremor.
And critically, damage to the lateral hemispheres strongly suggests the presence of the cerebellar cognitive affective syndrome.
Because that is the territory connected to the prefrontal and parietal cortices.
Mm.
The cerebellum is functionally localized not only for movement, but for mind. Hashtag tag tag outro.
This has been a true deep dive into the brain's massive hidden computational engine. We started with the astounding realization that the cerebellum contains 80 billion neurons, 80% of the entire CNS, organized into a perfectly uniform cortical matrix. We learned that the core computational concept is one of constant, precise regulation. Purkinje cells receive dual input, broad contextual mossy input distributed across the long parallel fibers, and the specific powerful error signal climbing input, and they use that dual signal to apply targeted GABAergic inhibition to modulate the constantly excited DCN output. It is a system built for meticulous, high-resolution tuning and refinement.
The immense level of detail we've covered, from the specific architecture of the cerebellar glomerulus, the planar geometry of the 15 million Purkinje cells, the crucial cross-wiring of the peduncles for ipsilateral control, to the precise division of the inferior olive subdivisions projecting to specific zebra bands. It all points to a dedicated, high-fidelity system built for optimization.
It really does.
The historical shift from viewing the cerebellum merely as a motor coordinator to recognizing its essential involvement in higher-order function, encapsulated in the cerebellar cognitive affective syndrome, is the direct culmination of this anatomical understanding. The cerebellum acts as a universal processing engine, using its repetitive circuitry to ensure that every system it connects to, be it a descending motor command or a complex abstract thought sequence, is executed with maximal efficiency and minimal error.
And since the cerebellum is constantly adapting its synaptic strength through LTD and LTP based on those specific potent error signals delivered by the climbing fibers, we want to leave you with this final provocative thought to mull over. If the cerebellum's sole purpose is to minimize the gap between what you intend to do and what you actually achieve, then how much of our perceived reality, our seamless ability to speak, catch, reason, and act, is simply the silent, tireless, precise inhibitory work regulated by those 15 million Purkinje cells, constantly editing and perfecting the script of our lives? The cerebellum truly is the brain's ultimate tireless editor.